guys, so we'll start today's session. I'll share my screen and we can start today's session. You can let me know if my screen is visible and if I'm audible. I'll share my screen. Okay, so I hope I'm visible. My screen is visible and I'm audible. And in case you stop seeing the slides at some point, or if you are not able to hear my voice, please let me know. I'll reshare my screen. Okay. So good evening, guys. This is Aishi. I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay, Maharashtra, and today is the fifth live session of this course, Biochemistry, which is taken by Professor K. Subramaniam. And in these live sessions, basically, we discuss some of the previous year assignment questions, and we not only discuss the questions, we try to discuss the concepts behind those questions, and so that it's easier for us to solve similar type of other questions. Okay, and then finally we'll jump to the answer. Moving to the first question for today. Amylopectin is a polysaccharide of glucose and glucose with dash and dash linkages. Anybody?
1 to 4.
Okay. Any other answer? Okay, so it's basically amylopectin will have both 1,4 and 1,6 linkages. Okay, let's go to the basic of polysaccharides. So polysaccharides are basically formed when many monosaccharides are joined together, and these can mean these two components which are basically make up starch, like amylose and amylopectin, cellulose and glycogen, and which are basically polymers of D-glucose and differ by branching and their types of glycosidic bonds. Now let's come to starch. What is starch? Starch is the storage form of glucose in plants. It is found as insoluble granules in rice, wheat, potatoes, beans and cereals. It's composed of two kinds of polysaccharides, amylose and amylopectin. Okay, so if you see the amylose structure over here, this is an unbranched chain of amylose. So amylose is always unbranched. It has an alpha 1,4 glycosidic bond. Okay, amylose basically makes up about 20% of starch, and it consists of 250 to 4,000 alpha D glucose molecules connected by alpha 1,4 glycosidic bonds in a continuous fashion. Okay, and the polymer chains of amylose are coiled in a helical fashion. Okay, but if you see the structure of amylopectin, this is the structure of amylopectin. It has both alpha 1,6 glycosidic bond. Basically, this type of glycosidic bond helps them for the branching out. It also has alpha 1,4 glycosidic bond like amylose. So amylose is the unbranched unbranched component of starch, whereas amylopectin is a branched component. In amylopectin, we see both alpha 1,4 alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond. Okay. So amylopectin basically makes up as much of as 80% of starch. It's a branched chain polysaccharide. It contains glucose molecules connected by alpha 1,4 and alpha 1,6 glycosidic bond, as I said. And basically what happens is the starches hydrolyze easily in water as an acid to give smaller saccharides called dextrins, which then hydrolyze to maltose and finally glucose. Now, in our bodies, these are complex carbohydrates and they are digested by the enzymes amylase in saliva and maltase in the intestines. Okay, so they provide about 50% of our nutritional calories from the glucose obtained in digestion. So if you see amylose and amylopectin, in the presence of amylase will form dextrins, then maltose, and basically the various D-glucose units, because it's a polymer, so we'll get the monomeric form. If we talk about the other animal starch, which is glycogen, which is also a polymer of glucose, and it is stored in the liver and the muscle of animals. It's hydrolyzed in our cells at a rate that maintains the blood level of glucose and provides energy between the meals. It's similar to amylopectin but more highly branched. So the glucose units in glycogen are joined by alpha 1 for glycosidic bonds with branches attached by alpha 1 cis glycosidic bonds that occur every 10 to 15 glucose units. Okay. Cellulose is the major structural unit of wood and plants. It's a polysaccharide of glucose units in unbranched chains with beta 1 for glycosidic bonds. It cannot form hydrogen bonds with water, right? So even if you try to mix it with water, it will still remain insoluble. It cannot dissolve in water, and it gives a rigid structure to the cell walls in wood and fiber, and it is more resistant to hydrolysis than other starches, and it cannot be digested by humans because humans cannot break down beta 1 for glycosidic bonds. Okay, so that is why this cannot be digested by humans as well. So this is cellulose. The polysaccharide cellulose is composed of glucose units connected by beta 1 for glycosidic bonds. Okay, now if you see this entire structure, which we can call the concept map, you see that carbohydrates can be categorized into monosaccharides, disaccharides, and polysaccharides. Okay, mostly monosaccharides, basically it's pretty much self-explanatory from the name. There'll be only one glucose sugar unit, basically one, and it will not have any glycosidic bonds that include glucose, galactose, fructose. They can be drawn as Fischer projections in the cyclic structures. Howard structures. We have talked about this in one of the previous classes. When the monosaccharides bond glycosidic bonds, they'll form disaccharides. So, two sugars. There'll be maltose, lactose, and even sucrose. Okay, and for polysaccharides will be obviously many number of sugars. They are polymers of glucose. They can be found in plants such as amylose, amylopectin, cellulose, and in animals the animal starch is known as glycogen. Okay, so the question was, coming back to the question, amylopectin is a polysaccharide of glucose with 1,4 and 1,6 linkages. So both 1,4 and 1,6 will be correct. Had it been amylose, only 1,4 would have been correct. Okay, moving to the next question for today. Which of the following is or are false? Number statement 1, the primary sequence of a protein dictates its secondary and tertiary structure. Statement 2, the pH scale is exponential. Statement 3, the N terminus of a protein is positively charged. Statement 4, upon forming a phosphorylated bond, the phosphate group in a polynucleotide can dissociate to give OH minus, and hence the polymer polynucleotide chain is a base in solution. And number 5, nucleotide is a repeating monomer having a nitrogenous base, a hexose sugar, and a phosphate group. Anybody?
Statement 1 and 2 are incorrect.
Sorry, statement. Can you repeat once?
The primary sequence of protein dictates its secondary and tertiary.
It's incorrect. Okay. And other statements? Which are statements incorrect? Okay, I'll wait for one more minute. Let's think about all the statements and then we'll start discussion. Okay. So we'll talk about the false statements. So these are the accepted answers. These are the false statements. Okay, as you can see over here. Now let's go with the sentences one statements one by one and decide which one is true and which one is false. Okay. Okay. So if we talk about nucleic acids, okay, we know the general structure of a nucleotide contains a nitrogen containing base. You can see over here a sugar, a phosphate group. Okay. Now there are two types of nucleic acids: the deoxyribonucleic acid DNA, which may contain several million nucleotides, and then ribonucleic acid or RNA, which may contain several thousand nucleotides. Now both are unbranched polymers of repeating monomer units known as nucleotides. And each nucleotide has three components: a base that contains nitrogen, a 5-carbon sugar, and a phosphate group. Okay. Now the bases in DNA and RNA are derivatives of the heterocyclic amines pyrimidine or purine. Pyrimidines with a single ring containing 2 nitrogen atoms, okay, and purines with 2 rings each containing 2 nitrogen atoms. So the H plus acceptors are the nitrogen atoms in each base. Okay, so in bases of DNA, if we talk about DNA, we have cytosine, thymine, adenine, and guanine. In DNA, the purine bases with double rings are adenine and guanine, and the pyrimidine bases with single rings are the cytosine and thymine. Whereas in RNA, the only difference with respect to the identity of bases will be that in RNA we don't really have thymine, we have uracil in place of that. So the purine bases with double rings are adenine and guanine, and the pyrimidine bases with single rings are cytosine and uracil. Now the 5-carbon sugar are the pentose sugars. So the 5-carbon sugar in RNA is ribose and in DNA is deoxyribose with no oxygen atom on C2 prime. So the 5-carbon sugar has carbon atoms numbered with primes to distinguish them from the atoms in the bases. If you see, these are the pentose sugars in RNA and DNA, ribose in RNA and deoxyribose in DNA. Now, what is a nucleoside then? A nucleoside is basically composed of a nitrogen containing base and a sugar, either ribose or deoxyribose, and it has a base linked by a beta N glycosidic bond to the C1 prime of a sugar. Can be ribose, if you can see over here, ribose or deoxyribose. This is how the condensation reaction will take place, and this is the how the base is linked by the beta N glycosidic bond. Now a nucleotide has a phosphate group attached to the pi 5 prime OH group of a nucleoside, and the addition of this phosphate to the nucleoside forms a nucleotide. So once the phosphate group joins the nucleoside, we get the nucleotide. Correct? So adenosine monophosphate, deoxyadenosine monophosphate, then guanosine monophosphate, deoxyguanosine monophosphate. All of these structures you can see over here. These are nucleotides of RNA and DNA. Cytidine monophosphate, deoxycytidine monophosphate, uridine monophosphate, or then deoxythymidine monophosphate. So uridine monophosphate, we don't really have deoxyuridine monophosphate because we cannot see uracil in DNA. And for thymidine, we don't see any thymidine monophosphate because we don't see thymidine in RNA. It's deoxythymidine monophosphate. So this is summary of the components in DNA and RNA. We have the bases A, G, C, T, and RNA we have A, G, C, U. The sugar will be deoxyribose in case of DNA and ribose in case of RNA. The nucleoside will contain base plus deoxyribose in DNA, base plus ribose in RNA. The nucleotide will contain base plus deoxyribose plus phosphate. In RNA it will contain base plus ribose plus phosphate. And the nucleic acid will be polymer of the deoxyribonuclease ribose nucleotides in DNA and polymer of ribose nucleotides in RNA. Now it is seen that every name of whenever we see a nucleoside, adenosine, so guanosine, so all these containing purine ends will end with osine, and the names of all the nucleoside that contains a pyrimidine ends with idine. Okay, thymidine, uridine, all of that. So the names of DNA nucleosides and add deoxy to the beginning of their names, and the corresponding nucleotides in RNA and DNA are named by adding monophosphate to the end of the nucleoside name. That is basic. That is the basic nomenclature of this system. So if you see the RNA, DNA nucleotides and nucleosides, these are the nucleotides and nucleotides in DNA and RNA. Okay. Now, what are AMP, ADP, and ATP? So, whenever we add a phosphate group to AMP, it forms a diphosphate ADP, and whenever we add a phosphate group to ADP, it forms a triphosphate ATP. So, if you see over here, phosphate then adenosine monophosphate, condensation will give ADP, and then adenosine triphosphate. Okay. Now, amino acids are the building blocks of proteins. They contain a carboxylic acid group and an amino group on the alpha carbon, the carbon adjacent to the CO, C double bond O functional group, because they have both have a weak acid and weak base present. So they actually exist as a salt. Okay, and each amino acid contains a different side group, R group. Okay, so obviously the R group will vary for different amino acids. The R group is what gives the amino acid its particular property, its particular, structure, particular conditions. All of these are provided by the R group. Everything else in an amino acid should be same for the different amino acids, but the R group, the side chain group, changes its entire functioning, changes its entire behavior. So this is structure of most amino acids. We have the side chain R, we have the amino group, and we have the alpha carbon, and we have the carboxylic acid group. So apart from the R group, all the three other components are pretty much the same in all amino acids. That's the common thing. Okay. Now, unlike most organic compounds, amino acids are soluble in water, have either very high melting points, or decompose upon heating. They act like more like inorganic materials than organic compounds. Now, when R is equal to H, the molecule is called glycine. When R is equal to CH, the molecule is called alanine. So, glycine is the simplest amino acid that we see in our see here. So, glycine, if you see, the R group is basically only hydrogen. Okay, so if you see, there are two similar components attached to the alpha carbon, two H groups attached, which is why this is this lysine does not really show chirality. Okay, so glycine is the simplest amino acid with H as the R group. Okay, so some R groups can be polar, some can be less polar, and so still some can be non-polar. So the polarity of these groups will affect the properties of the amino acids when they are combined to form proteins. Now these are the polar amino acids. If you can see, all of these structures are pretty much polar. Then we have the less polar, comparatively less polar, and then we have the comparatively non-polar amino acids over here. These are also polar amino acids. They are polar acidic amino acids, glutamic acid and aspartic acid. Now, what are essential amino acids? Essential amino acids are amino acids that are not produced by the body, but they are. That is why they are essential in your diet, because they are needed by the body, but the body does not produce them. So you need to have them in some way or the other. That is what we call essential amino acids. Now, what is a zwitterion? A zwitterion has an equal number of NH3 plus and CO minus groups, and it forms with the H group when the H from the COH in an amino acid transfers to the NH2 group. So this is glycine. The zwitterionic form of glycine will be this: O minus NH3 plus. Now the isoelectric point is the pH at which the amino acid has an overall zero charge. The isoelectric points of amino acids range from 2.8 to 10.8. Glycine with an isoelectric point of 6 exists as a positively charged species at a pH of below 6. This is the zwitterionic form that assess H plus, and then we see the positively charged ion. In solutions that are more basic than 6, the solutions with a pH of greater than 6, the NH3 plus will lose a proton and the amino acid will have a negative charge. Okay, so for example, glycine with a isoelectric point of 6 has a negative charge in solutions that have a pH above pH 6. Aspartic acid, which is an acidic amino acid, has as a isoelectric point of 2.8. So it is neutral salt at pH 2.8. Forms negative ions with charges -1 and -2 at pH values greater than pH 2.8. Okay. So according to the isoelectric points, we can basically figure a lot of things out about these amino acids. Now, why are the isoelectric points so important or significant? Why are we actually studying about them? So, in electrophoresis, an electric current is used to separate a mixture of amino acids. The positively charged amino acids move towards the negative electrode. The negatively charged amino acids move towards the positive electrode. And an amino acid at its isoelectric point does not migrate. The amino acids are identified as separate bands on the filter paper or thin layer plate. They provide a means of separating and identifying them. Now, this is a mixture of amino acids, and if you can see, if we consider a mixture of the amino acids glycine, valine, and aspartic acid at pH 6.0, that is subjected to electric voltage, which is which? Depending on the you can get to know the PI of these amino acids, PI or the isoelectric point of these amino acids, and from that we can try to figure out this type of question. Now, a positively charged positive species having pH lesser than isoelectric point move towards the negative electrode, whereas the negatively charged species having a pH of greater than uh, isoelectric point moves towards the positive electrode, a species with no net charge. And like a species with no net charge will not migrate, but a negatively charged species will move towards the positive electrode. Okay, so if you can see, lysine is a positively charged species, aspartic acid is a negatively charged species. Okay, so at pH 6, aspartic acid exists as this, and at a pH of 6, lysine exists as this. Now, all amino acids are chiral except glycine, which has two H atoms attached to the alpha carbon. All amino acids have the same relative orientation of groups in space. The other orientation is essentially not present in living organisms. It's the L orientation that all amino acids possess, in contrast with sugars, which have the D absolute orientation. Again, we talked about about this in one of the previous sessions. Now we have talked about amino acids. How do the peptides form? How do the amino acids come together, get linked up, and form the peptides? Okay, so the formation of peptides require the formation of an amide linkage. Okay, if you see over here, glycine and alanine have come together, and there is a peptide bond in between them, and the two amino acids have joined together. Okay, now a dipeptide is named with a YL ending for the N terminus amino acid in sequence. So the full amino acid name of the free carboxyl group at the C terminal. So if you can see, from alanine, alanine, from glycine, glycine, and from serine, serine only. So this is the structure of your peptide. We have alanine, glycine, and serine, these three, and we have one N terminus and one C terminus. Okay. So the primary structure of a protein is a particular sequence of amino acids that form the backbone of a peptide chain or proteins. Okay, so the primary structure of proteins will always be the sequence of amino acids. Okay, basically the letters of the it's basically the primary sequence of the amino acids that will form the backbone of any kind of protein. Now, the non-peptides oxytocin and vasopressin have similar primary structures, differ only in the amino acid position 3 and 8. Insulin was the first protein to have its own primary sequences determined. So, it is a primary structure of two polypeptide chains linked by disulfide bonds, and a chain with 21 amino acids and a B chain with 30 amino acids. Okay, so that's the first thing that we remember about insulin. It's a first protein because that was the first protein in which the primary structure was determined fully. Now, the primary structure of protein is the particular sequence of amino acids that form the backbone of the peptide chain of protein. The secondary structures of proteins indicate the three-dimensional spatial arrangements of the polypeptide chains. Now, the secondary structure, even though it has many forms, turns, loops, etc., but still we'll discuss the most popular ones: the alpha helix, which is a coil shape held in place by hydrogen bonds between the amide groups and the carbonyl groups of the amino acids along the chain hydrogen bonds between the hydrogen atom of an NH group and the O or the oxygen of the C double bond O of the fourth amino acid down the chain. So that is how alpha helix is formed. But in case of beta plated sheet, it is a secondary structure that consists of polypeptide chains arranged side by side. It has hydrogen bonds between the chains and has R groups above and below the sheet is typical of fibrous proteins such as silk. Okay. Then comes to the triple helix. Triple helix basically consists of three alpha helix bundles or chains woven together between large amounts of glycine, proline, hydroxyproline, and hydroxylysine that contain OH minus groups for hydrogen bonding. And it is apparently found in various kinds of collagen, connective tissue, skin, tendons, and cartilage. The primary structure of a protein is the particular sequence of amino acids that form the backbone of the peptide chain or the persona. Okay, so obviously, the primary structure will always be the sequence of the amino acids that will form the backbone of the peptide chain of protein. Okay, so the nature of bonding can be based on hydrophobic interactions between the nonpolar groups, can be hydrophilic interactions between the polar ionized groups and water on the surface of the tertiary structure. They can be involved in salt bridges, ionic interactions between ionized acidic and basic amino acids. They can occur between hydrogen, oxygen, and nitrogen, and they are disulfide bonds, strong covalent links between sulfur atoms of two cysteine amino acids. Secondary structure of proteins indicate the three-dimensional spatial arrangements of the polypeptide chains. So if you can see the image over here, this is the secondary structure of tubulin, structure of a protein. Now, the tertiary structure of a protein gives specific three-dimensional shape to the polypeptide chain, including interactions and cross-links between different parts of the peptide chain. And the tertiary structure is basically stabilized by different types of forces or interactions, such as hydrophobic and hydrophilic interactions, salt bridge hydrogen bonds, and disulfide bonds. If you talk about globular proteins are transported contact spherical shape, uh, carry out synthesis, transport, and metabolism in the cells, and are proteins such as myoglobin stores and transpose oxygen in muscles. So we have talked about the different sequences, different levels of protein structure. So the quaternary structure is also there. Basically, it's a combination of two or more tertiary units, and it's stabilized by the same interactions found in tertiary channels. So hemoglobin consists of two alpha chains and two beta chains with heme groups in each unit that pick up oxygen for transport in the blood to the tissues. Summary of the protein structure. So now, if we come to denaturation involves the disruption of salts in the secondary, tertiary, and quaternary protein structures. Okay. Now there are heat and organic compounds that break apart hydrogen bonds and disrupt metabolic hydrophobic interactions, acids, and bases that break H bonds between polar R groups and disrupt ionic bonds, heavy metal ions that break with S, and S bonds to have form heavy metal sulfides, agitation such as whipping, and that stretches peptide chains until bond breakage. Whereas denaturation basically can be seen when egg is boiled, the skin is wiped with alcohol, and the heat is used to catheterize blood vessels, instruments, and sterilize are in all this happens in the autoclave. So this is the classification of some proteins and their functions. If you see the structural proteins, what are the functions it provides, structural components. Also for contractile, we have the muscles which contract muscle fibers. Then we have transport, which basically carry essential substances throughout the body, nutrients. Hemoglobin transports oxygen, and lipoproteins transport lipids. Then we have seen storage nutrients, that's Casey. Milk protein, like casein stores protein in milk, ferritin stores iron in the spleen and liver, insulin regulates blood pressure, blood glucose level, growth hormone regulates body growth, and sucrase cells catalyze the hydrolysis of sucrose. So it's a schematic of basically the basic proteins and their functions. Now, a relatively new group of diseases are proteins such as called prions. So, this has come into the hotspot now. So, in the non-infectious form, the tail is a random coil, and once ingested, the tail can get folded into a beta-pleated sheet. It now becomes an infectious agent and has devastating effects on the brain and spinal cord, and some of it gets into the bloodstream without getting metabolized. The protein appears to be found in brain spinal cord acid. Okay. So does anybody have any question till now? There's some lagging issue that is happening because of my laptop. I don't know why. I'll just reshare and come. Because of the lagging issue, I'll come in 5 minutes. Anybody has any question, you can ask now. I'll reshare and come. Yeah, let me reshare. Okay. So we talked about the one of the statements about the primary structure basically dictating the secondary and the tertiary structure of protein. Yes, it's true. The primary structure can dictate the other level structures of proteins, because the primary structure means basically you're dictating the primary structure basically means the sequence of amino acids, right? So depending on the sequence of amino acids, where the hydrophobic amino acids are, where the hydrophilic ones are, polar ones are, you can basically figure out the folding pattern which makes up your tertiary and quaternary structure. So the folding pattern can be understood or determined from the primary sequence or primary structure. So yes, that statement is true, it's not false. Now moving on to the other statements. Before that, let's talk a little bit about acid and base. What are these and what is the classification criteria? So basically, acids and bases are classified into two categories based upon their strength, that is the degree to which they form ions. So if we say it's a weak, strong acid or strong base, that means it's substance that completely dissociates or ionizes to produce a maximum number of ions when dissolved in H2O. All other acids and bases are classified as weak. They produce few ions when dissolved in water. That means the dissociation is not entirely proper. Correct? So all the formula units dissociate separate ions. So dissociation is complete. For example, if you see this NaOH, Na plus and OH minus. So strong base. So what can be a strong acid which completely ionizes in water and no molecules will exist in the water solution. So for HCl and H2O, we can see H2O plus and plus Cl minus. Okay, so in general, the weaker the HA bond, the stronger the acid. So the weaker the HA bond, the more will be the dissociation, right? So that will make the acid more and more stronger. If the HA bond is proper and HA bond is stronger, then the dissociation will be lower. That will make the acid a weak one. Okay. So for example, the highly electronegative fluorine, it basically creates a very weak acid with hydrogen because it has such a strong attraction for its shared electrons. The Ha bond is not weak at all. So that is why it makes a weak acid, because its dissociation is not dissociation is not proper. It's not complete. Okay, that's a more right word. So dissociation is not complete. So that means because of its such strong attraction for its shared electrons, it makes up a very weak acid. So weak acid will be acid in which almost all the molecules remain as molecules when they are placed in solution. They are not dissociated. For example, acetic acid, phosphoric acid and carbonic acid. So the molecular structure of a weak acid determines the extent to which the acid ionizes in water. Now weak base. So weak base will be base in which the most of the molecules do not react with water to form ions. For example, ammonia, aluminum hydroxide and all of that. Weak acids and bases. So weak does not mean unimportant. Okay. Due to the wide variety of structures possible, there is a wide range of weakness to this group. And they are considered to be weak because there is only partial ionization of these acids and bases into hydronium or hydroxide ions. Now, weak and strong, the strengths of acids and bases, and dilute and concentrated, what are these terminologies for? To measure the concentrations of solutions. So, both strength and concentration determine the behavior of the solution. So, if this is a pH scale, we have more acidic, more basic, and a neutral in the middle. Okay. So, the pH scale basically means power of hydrogen. It measures the concentration of H3O plus, H plus ions, H plus in solution, and it is used to make the wide range of possible concentrations easier to work with. So pH is a mathematical scale in which the concentration of hydronium ions in a solution are expressed as a number from 0 to 14. Basically, it's a logarithmic scale of 10 to the power 0 to 10 to the power minus 14. So the value of pH is the negative of the exponent. Okay, that is 0 to 14. So if H plus ion concentration is 10 to the power minus 6 molar, the pH will be 6. So if pH is 12, then H plus will be 10 to the power minus 12 molar. Now pH can be used to determine POH, that is the power of hydroxide. So if pH is equal to 12, then POH will be 2. So pH basically will be the difference between 12 and 14, the higher end of the pH scale minus the pH value given. So pH plus POH will be equal to 14. So if pH is 12, then OH minus will be 10 to the power minus 2 molar. Okay. Now the pH value can be calculated on calculator using the logarithmic function obviously. So pH will be minus log H plus concentration, where H plus will be your hydrogen ion concentration, whereas POH will be minus log OH minus H minus, where OH is the hydroxide ion concentration. And pH plus POH will always be 14 at the temperature of 25 degrees Celsius. Okay. Now measuring pH. So indicators will register different colors at different pHs. Like this is an indicator paper, and when you add a few drops of your solution into it, depending on the pH of the solution, it will show different colors. So, and now pH meters also measure exact pH, but if you're talking about indicator, it will not be the exact pH, or based on the color change, and based on the color chart that you have with you, you can predict the range. Okay. pH greater than 7 is obviously basic. 7 is the neutral pH. pH greater than 7 basic, and pH 7 which is neutral, not acidic or basic if it's 7. If the pH is less than 7, it's acidic. Okay. So as the pH increases above 7, the concentration of hydroxyl ion increases and the concentration of hydronium ion decreases. Okay. So every drop in pH is 10 times increase in H plus. Okay. So dropping from pH 5 to pH 4 means that the H plus of pH 4 is 10 times greater. Okay. So every 1 unit increase in the pH will be a factor of 10 increase in the OH minus in the concentration. So you can figure out how fascinating is that. Every 1 unit increase will be in the pH, but a 10 factor rise or increase in the OH concentration. Okay. So in a neutral solution, the concentration of OH minus and the concentration of H2O plus will be equal. Now, if we talk about some common substances which are as mentioned over here, what are the pH of these common substances? Like lemon juice, it's a very acidic mixture. That's why the pH is very acidic, 2.3. Then we have milk, pH 6.4. We have ammonia, 11.9. All of these. Now let's talk about acid-base indicator ranges. Now there are some colored solutions which we call indicators that have different colors at different pHs, and they give approximate pH by comparing the color to a standard chart. So we have a standard chart where we know which color points to which pH. Okay. So these are the basic function of acid-base indicators. Now neutralization reaction. So what is the neutralization reaction? It occurs when an acid reacts with a base to form a salt and water. Salts are crystalline solids that may or may not be soluble in water. Okay. So H2SO4 plus 2NaOH will give you Na2SO4 plus H2O. Salts contain the positive ion of a base and the negative ion of an acid. Some salts called acid salts contain hydrogen, that is NaHSO4 or K2PO4. These are the acid salts. What is the pH scale? There was one question about pH scale. So the ion product of water, Kw, is the basis for the pH scale. The pH scale is a convenient way of designating the concentration of H plus and OH minus in solution in the range between 1 molar H plus and 1 molar OH minus. So the pH is defined by the equation pH is equal to minus log H plus ion concentration. So precisely a neutral solution occurs when H plus is equal to OH minus, and thus when pH is equal to 7. Okay, a solution in which the H plus will be greater than OH minus will be acidic and have pH values less than 7. Solutions in which H plus will be less than OH minus, it will be basic because OH minus is more and will have a pH of value greater than 7. Now if we talk about other some common liquids for the pH values, we see the pH values of some common aqueous fluids are shown here. So the pH scale is logarithmic, and solutions that differ by 1 pH unit actually differ by 10-fold in H plus ion concentration. The H plus ion of a solution of known pH is calculated by rearranging the pH equation to its exponential form. So the pH of biological solutions is carefully controlled because pH strongly affects the structure of and activity of biological macromolecules, that is enzymes. Okay, so we have talked about the pH range. That was one of the statements there. Coming back to the protein structure and function, we have talked about this, but let's talk a little bit more. So we have talked about the 4 levels of protein structure, the hierarchy in protein structure and how it happens. So this is a detailed discussion of each of these levels is presented in the next 2 slides also, and experiments have even shown that the final 3D tertiary structures of a protein ultimately is determined by the primary structure. See, so there are experiments, it's not only a theoretical proof, there are experiments that show that the primary structure will knowingly or unknowingly determine its tertiary structure. The 3D fold of the protein determines its function basically. As I said, the primary structure of a protein refers to the amino acid sequence. Now, amino acids in the peptides and proteins, typically 20 to 1,000 amino acids, are joined together by peptide bonds or amide bonds between the carboxyl and the amino groups of adjacent amino acid. Now, the backbone of all proteins consists of, as we said, N, C alpha, R side chain, and C double bond O repetitive unit. So only the R group side chains vary. By convention, the protein sequences are written from left to right from the proteins N to C terminus. Okay. So the average yeast protein contains 446 amino acids. Because the average molecular weight of an amino acid is 110 daltons, the average molecular weight of the yeast protein will be this. Okay. Coming to the secondary structure, which basically the most popular ones, alpha helix and beta pleated sheet. They refer to short-range periodic folding elements that are common in proteins, and these include the alpha helix, the beta sheet, and turns. So in the alpha helix, the backbone adopts a cylindrical spiral structure in which there are 3.6 amino acids per turn. So the R groups point out from the helix and mediate contacts to other structural elements in the folded protein. Now the alpha helix is stabilized by hydrogen bonds between backbone carbonyl oxygen and amide nitrogen atoms that are oriented parallel to the helix axis. H bonds occur between residues located in the N and the N plus 4 positions relative to one another. Now in the beta pleated sheets, each beta strand adopts an extended conformation. Now beta strands tend to occur in pairs or multiple copies in beta sheets that interact with one another by H bonds directed perpendicular to the axis of each strand. Now carbonyl oxygens and amide nitrogens in the strands form the H bonds, and strands can orient anti-parallel or parallel to one another in beta sheets. So the R groups of the side chains of every other amino acid point up or down relative to the sheet. So most beta strands in proteins are 5 to 8 amino acids long, and beta turns consist of 3 to 4 amino acids that form tight bends, and glycine and proline are common in turns. Longer connecting segments between beta strands are called loops. Now if we talk about tertiary structures, it basically refers to the folded 3D structure of a protein. It is also known as the Lady structure or active conformation. Tertiary structure mostly is stabilized by non-covalent interactions between secondary structural elements and other internal sequence regions that cannot be classified as a particular type of secondary structure. So the folding of proteins is thought to be driven by the need to place the most hydrophobic regions in the interior out of contact with water. As I was saying, why does the primary sequence determine the tertiary structure? Because if there are hydrophobic amino acids in the sequence, which is obviously dictated by the primary level of structure, if there are such hydrophobic regions in the basically in the interior, that should not be in contact with water. Okay, they should be during the folding, they should be taken inside in the interior of the protein. Now the structures of hundreds of proteins have been determined by techniques such as X-ray crystallography and NMR, and different methods of representing structures are shown over here. Now, what are secondary structures? Motifs. Secondary structure motifs are evolutionary conserved collections of secondary structural elements which have a defined conformation. They also have a consensus sequence because the alpha because the amino acid sequence ultimately determines structure. So, a given motif can occur in a number of proteins. In the number of proteins where it carries out the same or similar functions. Some well-known examples such as the coil, EF hand, helix, loop helix, and zinc finger motifs. So they are also illustrated over here. These motifs typically mediate protein-protein association, calcium DNA binding, and DNA or RNA binding respectively. Okay. Now quaternary structures. Basically, if the protein is a multi-subunit protein, okay, so if there are different subunits that need to come together for the protein to be functional, if it's a multimeric multi-subunit structure, they'll have another level of structural organization known as quaternary structure. So quaternary structure refers to the number of subunits, their relative positions and contacts between the individual monomers in a multimeric protein. So the quaternary structure of the trimeric hemagglutinin surface protein of influenza virus is shown here. The tertiary structure of a hemagglutinin monomer is shown here. Okay. Now, modular domain structure of proteins. So, domains are independently folding and functionally specialized tertiary structures units within a protein. Okay, so the respective globular and fibrous structural domains of hemagglutinin monomer are illustrated here. So, domains such as the EGF domain may also be encoded within a single polypeptide chain. Now, domains still perform their standard functions, although fused together in a longer polypeptide. And the modular domain structure of many proteins has resulted from the shuffling and splicing together of their coding sequences within the longer genes. Now, what is a supramolecular structure then? So, in many cases, multimeric proteins achieve extremely large sizes, that is tens and hundreds of subunits, and such complexes exhibit the highest level of structural organization known as supramolecular structure. The examples can include the mRNA transcription pre-initiation complexes, ribosomes, proteasomes, and spliceosomes. So typically, supramolecular complexes function as macromolecular machines in reference to the fact that the activities of the individual subunits are coordinated in the performance of some overall task. Now, if we talk about the evolution of protein families, though genome sequencing and classical gene cloning approaches, the sequences of an enormous number of proteins have been compiled. The comparison of sequences shows that the most proteins belong to larger families that have evolved over time from a common ancestor protein. Okay, so proteins that have common ancestor are called homologs. The members of a protein family often show 30% sequence ID have a common 3D fold and usually perform closely related functions. Okay, so if we talk about the structure of the globin proteins, they are mostly alpha helical secondary structures, and the similar folds of the globins can be readily seen by comparing such as the beta chain of hemoglobin, myoglobin, and lake hemoglobin. The closely similar structures of mammalian myoglobin and hemoglobin beta subunit might be expected, but the resemblance of the distantly related plant like hemoglobin is striking. So the comparison of the sequences of the members of protein families has brought to light the fact that amino acids within a given class exhibit a large degree of functional redundancy. So in this regard, the three proteins discussed over here will be exhibiting less than 20% identity in the sequences, yet they have the same structure. Lastly, in the hemoglobin, two different globin chains are combined to form a multi-subunit protein. So if you talk about a summary of the protein folding, so many experiments have shown that proteins can spontaneously fold from an unfolded state by their folded native state. So this proves that the amino acid sequence contains enough information to specify tertiary structure. So bonds within the peptide backbone seek out different possible conformations as the final tertiary structure is achieved. Okay, now folding tends to occur by successive conformational changes leading to secondary and then tertiary structural elements. So the native conformation of a protein typically is its lowest free energy and therefore most stable structure. So the unfolded or denatured conformation of a protein can be generated by heating or treatment with certain organic solvents. Okay, let's now go into this. If we talk about enzyme catalysis, so enzymes are proteins basically that catalyze chemical reactions within living organisms. So enzyme catalyzed reactions typically are highly specific in the rate enhancements of 10 to the power 6 to 17 are common. So in an enzyme catalyzed reaction, the reactants, the substrate is converted into the product. So like all catalysts, enzymes are not consumed in the reaction. Further, they do not change the properties of the reaction, only its rate. So rate enhancement is achieved due to the fact that enzymes are most complementary to the transition state structure formed in the reactions. This results in stabilization of the transition state and lowering of the activation energy barrier. Okay, so the transformation of a substrate to the product can occurs in the active state of an enzyme. Now the active site can be subdivided into a catalytic site wherein amino acids that catalyze the reaction reside in a binding pocket that recognizes specific features of the substrate, conferring specificity to the enzyme substrate interaction. So, a schematic model for an enzyme catalyzed reaction is shown here. So, the kinetic equation describing the reaction will be E plus S, ES, and E plus P, which is enzyme plus product. Reaction coordinate diagram showing the binding and catalytic states of an enzyme catalyzed reaction is shown here. So the velocity of an enzyme catalyst reaction, which is a maximal rate at high concentrations of substrate, we see the Vmax is achieved when all enzyme molecules are bound to substrate and engaged in catalysis. Okay, so these are basically two French mathematicians, Michaelis and Mentin, who got forever famous in the field of biology because of the Michaelis-Menten equation. So they developed a kinetic equation to explain the behavior of most of these enzymes. They showed that the maximal rate of an enzyme catalyst reaction depends on the concentration of the enzyme and the rate constant for the rate limiting step of the reaction. So the equation will be V naught equal to V max S by S plus Km. Now, Michaelis and Menten also derived a kinetic constant, the Michaelis constant, that is indicative of the affinity of most enzymes for their substrates. So, the lower the Km, the higher the affinity of the enzyme for the substrate. So, the Km happens to be the concentration of the substrate at which the reaction rate is half maximal. So, the concentrations of cellular metabolites usually are set near the Kms of the enzymes that carry out their metabolism. So, this allows the cells to respond to changes in the substrate concentration. Okay, so these are mechanisms of certain proteins. We don't get into that. So there are also some multifunctional enzymes. So most metabolic pathways occur via multiple enzyme catalyzed steps, such as like the rates of pathway reactions can be increased if the substrate and products of each step are channeled to the next enzyme in the pathway. So channeling is enhanced in multi-subunit enzyme complexes and by attachment of enzymes to scaffolds, or even fusion of the encoded enzymes into single polypeptide chain. Okay, won't go into all these details, but basically the question was, coming back to the main question. One second. Wait. Okay. Coming back to the main question. Yeah. So the main question was, the primary sequence of a protein dictates its secondary and tertiary structure. True. The statement is true. The pH scale is basically logarithmic. The N terminus of a protein is positively charged. That is true. Upon forming a phosphodiester bond, the phosphate group in a polynucleotide can dissociate to give OH minus, and hence the polymer polynucleotide chain is a base in solution. This is false. Nucleotide is a repeating monomer having a nitrogenous base, a hexose sugar, and a phosphate group. So that is true. So the rest, apart from, basically it's not true. Sorry, this is not true. This is false, because it's a repeating monomer having a nitrogenous base and a sugar. It can be a pentose sugar as well. Basically it's a pentose sugar, not hexose. It's a pentose sugar and a phosphate group. So apart from that fact, this statement would have been correct, but this statement is false. Okay, this statement is false, this statement is false, and this is only 1 and 3 are correct statements. Okay, so that was all about this question. We'll move on to the next question for today. So identify the correct classifications. Okay, anybody would like to answer? Anybody? Am I audible? Yeah, I'm audible. Anybody wants to answer this question? I'll give you a minute. You can think about it and answer.
Purine is adenine and
Purine is adenine and pyrimidine is uracil. Okay. Any other option?
purine is guanine.
Okay. Right. Anybody else? Yeah. So the accepted answers, the correct answers will be purine is adenine, correct? While the pyrimidine is uracil, which is found in RNA. Or purine is guanine and the pyrimidine is thymine, which is found in DNA. I talked about this also, but a little briefly. Let's go over it. So we have talked about nucleic acids and nucleotides. We know that the nucleotides have a variety of roles in cellular metabolism. They have the energy currency in metabolic reactions, the essential chemical release in the response of cells to hormones and other stimuli, and the structural components of a variety of enzyme cofactors and metabolic intermediates. So they are also constituents of the nucleic acids, deoxyribonucleic acid, DNA, and RNA, and the amino acid sequence of every protein in a cell, and the nucleotide sequence of every RNA specified by nucleotide sequence in genomic DNA. So segments of DNA specifying the synthesis of a functional protein or RNA product are called genes. So the storage and the transmission of biological information are the only known functions of DNA. RNAs have a broader range of functions. Ribosomal RNAs are structural and catalytic components of ribosomes, whereas messenger RNAs carry genetic information specifying the sequences of proteins. Transfer RNAs are adapted molecules that participate in translating the information in mRNA into a specific sequence of amino acids in a polypeptide. So there is also a wide variety of special function RNAs, including some such as the ribozymes, which has enzymatic activities. So if someone tells you that only proteins have enzyme activities, they are wrong, because there are certain RNAs also which can have enzymatic activity. They are called ribozymes. Now, coming to the structure of nucleotides, basically three components: nitrogen containing base, a pentose, and one or more phosphates. So in the absence of the phosphate groups, the molecule is called a nucleoside. So nitrogenous bases are derivatives of pyrimidine and purine. So the numbering of the ring atoms of pyrimidines and purines are illustrated over here. Okay. So the numbering of the pentose rings follows the normal convention, except that the carbon numbers in the pentoses of nucleotides and nucleosides are given a prime designation to distinguish them from the numbered atoms into the bases. So the base of a nucleotide is joined covalently at N1 of pyrimidines and N9 of purines in the N-beta glycoside bond to the 1 prime carbon of the pentose, and the phosphate is esterified commonly to the 5 prime carbon. Water is removed in the formation of the N-beta glycoside bond, as occurs in the O glycosidic bond formation. Okay. Now, what are the major bases of nucleic acid? Both DNA and RNA contain two major purine bases, adenine and guanine, and both nucleic acids also contain the pyrimidine cytosine and a second pyrimidine, that is thymine in case of DNA, uracil in case of RNA. So occasionally does thymine occur in RNA or uracil in DNA, only occasionally. So in some cases the names of the bases reflect the sources from which they originally were isolated. Guanine, for example, was first isolated from guano, which is apparently a bird manure, whereas thymine was first isolated from thymus tissue, hence the name thymine. So if we talk about the nomenclature of nucleotides and nucleosides, it's given over here. What are the different nomenclatures, how they are called when they are in the nucleoside form, and how they are called in the nucleotide form. Okay, So nucleoside and nucleotide are generic terms that include both ribo and deoxyribo forms. Also, ribonucleosides and ribonucleotides are here designated simply as nucleosides and nucleotides. Okay, and deoxyribonucleosides and deoxyribonucleotides as deoxynucleotides and deoxynucleosides. So both forms of naming are acceptable, but the shortened names are more commonly used. Thymine is an exception. Ribothiamidine is used to describe its unusual occurrence in RNA. Now, nucleotides have two kinds of pentoses. The decoding deoxyribonucleotide units of DNA contain 2' deoxyribose and the ribonucleotide units of RNA contain D-ribose. So in both the types of nucleotide, the pentoses exist in their beta furanose. That is a closed 5-membered ring form. The formation of the beta ribofuranose ring from the straight chain aldehyde form of D-ribose in a solution is illustrated over here. And the deoxyribose undergoes a similar interconversion in solution, but in DNA exists solely as beta 2' deoxyribofuranose. Now, deoxyribofuranose and ribofuranose rings in nucleotides can exist in 4 different puckered conformations, and in all cases, the 4 of the 5 ring atoms are nearly in the same plane. The fifth atom, the C2' or C3', is on either the same endo or the opposite side of the plane exo, related to the C5' atom. Okay, this is deoxyribonucleotides of DNA. And all nucleotides are shown in their free form as pH 7, the neutral pH. So deoxyribonucleotide units of DNA are usually symbolized as AGDC and sometimes as DA, DG, DT and DC. In their free form, the deoxyribonucleotides are commonly abbreviated as DAMP, DGMP, DC, DCCMP and DTMP. For each nucleotide in the figure, the more common name is followed by the complete name in the parenthesis. So all the abbreviations assume that the phosphate group is at the 5' position. Nucleoside portion of each molecule is shaded in light red over here, if you can see over here. Now the structure and names of the 4 major ribonucleotides of RNA are shown here, and all nucleotides are shown in their free form at 7 neutral pH. Now the ribonucleotide units of RNA are usually synthesized A, G, U and C, and in their free forms, the ribonucleotides are commonly abbreviated as AMP, UMP, GMP and CMP. And for each nucleotide in the figure, the more common name is followed by the complete name and the parenthesis. All abbreviations followed assume that the phosphate group is at the 5' position, and the nucleoside portion of each molecule is shaded in light red. Over shaded, you can see over that. There are certain minor bases also in both DNA and RNA. In DNA, the most common of these are the methylated forms of the major bases. Minor bases of many times occur in tRNAs. If the modification occurs directly on one of the ring atoms of the pyrimidine or purine base, the convention is to simply indicate the ring position of the substituent by its number, like 5-methylcytidine. The convention changes when the substituent atom is exocyclic, not within the ring structure. So in this case, the type of atom is identified and the ring position to which it is attached is denoted with the superscript as N6-methyladenosine. Now some adenosine monophosphates cells also contain nucleotides with phosphate groups in positions other than on the 5' carbon of the pentose ring. Okay. So for example, ribonucleoside 2', 3' cyclic monophosphates are isolatable intermediates, and ribonucleoside 3' and 2' monophosphates are end products of the hydrolysis of RNA by certain ribonucleases. So other variants are adenosine 3', 5' cyclic monophosphate, which is a very important molecule in some signal transduction pathways. Okay, so CRP is a very popular molecule in very cell signaling pathways. If you have read cell signaling, especially in blue salvets and all, you can see that these molecules are very important for any kind of signaling cascading pathways. Now, what are the phosphodiester linkages in the covalent backbone of DNA and RNA? So, the successive nucleotides in DNA and RNA are covalently linked through phosphate group bridges in which the 5' phosphate of one nucleotide unit is joined to the 3' hydroxyl group of the annexed, creating a phosphodiester linkage. So, the covalent backbones of nucleic acids consist of alternating phosphate and pentose residues, and the nitrogenous bases may be regarded as side groups joined to the backbone at regular intervals. So the backbones of both DNA and RNA are hydrophilic. The hydroxyl groups at the sugar residues form hydrogen bonds with water. The phosphate groups with the pKa near zero are completely ionized and negatively charged at pH 7. So the negative charges are generally neutralized by ionic interactions with positive charges on proteins, metal ions, and polyamines. So all the phosphodiester linkages in DNA and RNA have the same orientation along the chain, giving each linear nucleic acid strand a specific polarity and distinct 5' and 3' ends. So by definition, the 5' end lacks a nucleotide at the 5' position, and the 3' end lacks a nucleotide at the 3' position. Now, the covalent backbone of DNA and RNA is subjected to slow non-enzymatic hydrolysis of its phosphodiester bonds. So in vitro, RNA is hydrolyzed rapidly under alkaline conditions, but DNA is not. So this is because the 2' hydroxyl group in the ribose moieties of RNA is directly involved in the cleavage process. 2' 3' cyclic monophosphate nucleotides are the first products of the action of alkaline RNA and are subsequently hydrolyzed further to yield a mixture of 2' and 3' nucleoside monophosphates. Okay, if you can see the structures over here. Now, how do we represent nucleotides? The nucleotide sequences of nucleic acid can be represented as illustrated here for a segment of DNA with 5 nucleotide units. The phosphate groups are symbolized by its circle P's over here, and each deoxyribose is symbolized by a vertical line from C1' at the top to C5' at the bottom. The connecting lines between the nucleotides are drawn diagonally from the middle of the deoxyribose to the one of nucleotide to the bottom of the next. So some simpler representations of this pentadeoxyribonucleotide are these structures, and you know that the sequence of a single strand of nucleic acid is always written with the 5' end at the left hand side and the 3' end at the right hand side. So that's in the 5' to 3' direction. So short nucleic acid such as shown in this figure is referred to as an oligonucleotide. This term is generally applied to nucleotides of 50 or fewer residues. Longer nucleic acids are referred to as polynucleotides. Now there are obviously different forms of the nucleotide, the bases basically. So tautomeric forms of uracil. So free pyrimidine and purine bases may exist in two or more tautomeric forms depending on the pH. So uracil, for example, occurs in lactam, lactine, and double lactine forms depending on the pH. Certain tautomeric forms predominate at neutral pH, and these are the structures shown for the 5 common purines and pyrimidines in here. So these are the tautomers that are present in the bases in DNA and RNA. Now, all nucleotides bases absorb UV light, and nucleic acids are characterized by strong absorption at wavelength near 260 nanometer. So, in this figure, in the variation in molar extinction coefficient epsilon as a function of wavelength, the molar extinction coefficients at 260 nanometer are listed in the dash table. Okay, so spectra of the corresponding ribonucleotides and the deoxyribonucleotides are essentially identical. So, for mixtures of nucleotides, a wavelength of 260 nanometer is used for absorption measurements. Coming to the Watson and Crick base pairing, we started our session, one of the first sessions with this topic, the Watson and Crick base pairing in DNA. So the functional groups of pyrimidines and purines are ring nitrogens, carbonyl groups, and exocyclic amino groups. So hydrogen bonds involving the amino and the carbonyl groups are the most important mode of interactions between two complementary strands of nucleic acid. The most common hydrogen bonding patterns are thus defined by Watson and Crick, in which A bonds specifically to T and G bonds to C. So these two types of base pairs predominate in double-stranded DNA and RNA, and the tautomers shown here. And the tautomers shown are responsible for these types of base pairs. So if this it is a specific pairing of bases that permits the duplication of genetic information in DNA. So basically, the model for the structure of DNA was proposed first by Watson and Crick in 1953. Their model was based on a number of pieces of information that were available at the time about the composition of DNA and the X-ray diffraction properties of DNA fibers. So most importantly, X-ray diffraction properties of DNA fibers. Okay, and the studies of DNA fibers were performed by Rosalind Franklin, who was deceived of the Nobel Prize, who did not get that much recognition at that time, and who actually passed away in a few years. So, Nobel Prize was not given posthumously at that time. So basically, Rosalind Franklin's X-ray diffraction data showed that the DNA molecules are helical and exhibit two periodicities repeating along the length of the fiber, a primary repeat of 3.4 angstrom and a secondary repeat of 34 angstrom. So in addition, Edwin Shargaf and colleagues had shown through DNA composition analysis that the number of T residues always equals the number of A residues. As a result, the sum of the purine residues equals the sum of pyrimidine residues is what comes out. So Watson and Crick then set about to develop a structure that was consistent with these two sets of data. Okay, so they then proposed the two helical DNA chains are wound around the same axis to form a right-handed double helix. They speculated that the two chains have an anti-parallel orientation, and this was later proven to be true. So the hydrophilic backbones of alternating deoxyribose and phosphate groups are on the outside of the helix facing the surrounding water. So the furanous ring of each deoxyribose is in the C2' endo conformation. The purine and pyrimidine bases of both the strands are stacked inside the double helix, and their hydrophobic and nearly planar ring structures are very closely together and perpendicular to the axis of the helix. Okay, so the base stacking will account for the 3.4 angstrom repeat along the length of the helix, and the secondary repeat of about 34 angstrom was accounted for by the presence of 10 base pairs in each complete turn of the double helix. So this was later modified to 10.5 base pairs per turn for DNA in aqueous solution, and in the Watson-Crick model, AT and GC base pairing is proposed based on the fact that these combinations of bases fit well inside the double helix. Finally, the offset pairing of the two strands creates a major and a minor groove on the surface of the duplex. So it should be noted that the double helix not only is stabilized by Watson-Crick base pairing between residues in the helix, but is also stabilized by base stacking interactions that remove the bases from the contact with water. So the features of the double helical model of DNA structure are supported by much chemical and biochemical evidence. Okay. This is double helical strand complementarity. The two antiparallel chains of double helical DNA are not identical in either base sequence or composition. Instead, they are complementary to one another. Wherever adenine occurs in one chain, thymine occurs in the other. Similarly, guanine occurs opposite cytosine in the two chains. Now, the model for DNA structure suggested by Watson and Crick also talked about a mechanism of genetic information transfer. Okay. The essential feature of the model is a complementarity of the two DNA strands in the double helix. So as Watson and Crick were able to see well before confirmatory data became available, this structure could logically be replicated by separating the two strands and synthesizing a complementary strand for each. So because nucleotides in each strand are joined in a sequence specified by the base pairing rules stated above, each pre-existing strand will function as a template to guide the synthesis of the complementary strand. Okay. Now, double helical DNA is a very flexible molecule, obviously. Considerable rotation is possible around several types of bonds in the phosphodeoxyribose backbone, and thermal fluctuation can produce bending, stretching, and unpairing or melting of the strands. Okay. Now, many significant deviations from the Watson-Crick DNA structure are found in cellular DNA, some or all of which may be important in DNA metabolism. So, we can know that these structural variations generally do not affect the fundamental properties of DNA, such as strand complementarity, anti-parallel strands, and the requirement for A, T, and GC base pairs. Structural variations in DNA are due to three things: the different possible conformations of the deoxyribose, rotation about the contiguous bonds that make up the phosphodeoxyribose backbone, and the free rotation about the C1' N glycoside bond. So, because of steady constraints, purines and purine nucleotides are restricted to two stable conformations with respect to the deoxyribose called syn and anti. So, pyrimidines generally are restricted to the anti conformation because of the steric interference between the sugar and the carbonyl oxygen at C2 of the pyrimidine. Okay, so this is a syn adenosine format, and this will be once it rotates, it can form the anti adenosine format as well. And if you can see this, it can form the anti-cytidine. So because of the steric constraints, the purines in the purine nucleotides are restricted to two stable conformations with respect to deoxydibose called syn and anti. Whereas pyrimidines are generally restricted to the anti conformation because of steric interferences between the sugar and the carbonyl oxygen of the at the C2 of pyrimidine. Now, what are the A, B, and Z forms of DNA? So, the Watson-Crick structure of DNA is also referred to as the B form DNA or bDNA. It is the most stable structure for a random sequence DNA molecule under physiological conditions and is therefore the standard structural reference in any study of the properties of DNA. Now, two structural variants that have been well characterized in crystal structures of the A and the Z forms of DNA. The properties of these forms is summarized in one of the tables in the next slide. So, in general, A DNA is a dehydrated form of DNA that may not occur in cells. A similar type of structure does occur in double helical RNA. There is evidence for some short tracks of Z DNA in bacterial and eukaryotic cells. So, these Z DNA tracks may play a role in regulating the expression of some genes or in genetic recombination. Okay, the DNA backbone of zDNA takes on a zigzag conformation. So certain nucleotide sequences fold into left-handed seed helices much more readily than others. So there are prominent examples of sequences in which pyrimidines alternate with purines, especially alternating C and G or 5-methyl C and G residues. So to form the left-handed helix in zDNA, the purine residues will flip to the syn conformation, alternating with pyrimidines in the anti-conformation. Now, DNA palindromes and mirror repeats. It is thought that other sequence dependent structural variations found in larger chromosomes may affect the weight function and metabolism of the DNA segments in their immediate vicinity. A rather common type of such DNA sequence is a palindrome. So, you know what a palindrome is, right? A palindrome is a word, phrase, or sentence that is spelled identically read either forward or backward. So, example is the word rotator. So, from even if you read from the backside, it will be rotator only. So the term is applied to regions of DNA with inverted repeats of base sequence having twofold symmetry over the strands or two strands of DNA. So such sequences have the potential to form unique DNA structures, and when the inverted repeat occurs within each individual strand of DNA, the sequence is called a mirror repeat. So mirror repeats cannot form the same types of structures such as DNA palindromes. Okay, so DNA palindromes are self-complementary within each strand and therefore have the potential to form hairpin because they can fold upon themselves. They can, because they have complementary sequences within themselves, they can fold to form a hairpin and cruciform structures. And sequences of these types are found in virtually every large DNA molecule and can encompass a few base pairs of thousands. So, extent to which palindromes occur in cruciforms in cells are not known. However, some cruciform structures have been demonstrated in vivo in E. coli. Self-complementary sequences cause isolated single strands of DNA or RNA in solution to fold into complex structures containing multiple hairpins. So if you see if these are complementary, it will form a hairpin-like structure, and this will form a cruciform-like structure. Now, what is triple helical DNA? Several unusual DNA structures involve 3 or 4 DNA strands, and the nucleotides participating in a Watson-Crick base pair can form additional hydrogen bonds, particularly with functional grooves in the major groove. For example, a cytidine residue, if protonated, can pair with the guanosine residue of GC base pair, and a thymidine can pair with the adenosine of an AT base pair. Okay. Now the N7O6 and N6 of purines are the atoms that participate in the hydrogen bonding of triplex DNA. So these atoms are then referred to as the Hookestein positions, and the non-Watson pairing is called Hookestein pairing after the discoverer Carl Hookestein. This type of base pairing allows the formation of triplex DNA. So the Hookestein base pairing will allow the formation of any kind of triplex DNA. Now, some triplex DNAs can contain two pyrimidine strands and one purine strand. So, others contain two purine strands and one pyrimidine strands. So, helical DNA containing two pyrimidine strands and one purine strand are shown over here. And the blue and white strands are antiparallel and are paired by normal Watson-Crick base pairs. And the third, all pyridine strand is parallel to the purine strand and paired through non-Watson-Crick hydrogen bonds. The triplexes are mostly stable at low pH because the CGC plus triplet requires a protonated cysteine In the triplets, the pK of this hydrogen is 7.5, which is raised considerably from its normal value of 4.2. And triplexes form most readily within a long sequences containing only pyrimidines or only purines in a given strand. Apparently, we have something called the tetraplex DNA also, where four DNA strands can also pair to form a tetraplex or quadruplex, and this occurs readily only for DNA sequences with a very high proportion of guanosine residues. So, the guanosine tetraplex or G tetraplex is quite stable over a wide range of conditions. Okay. If you talk a little bit about RNA structure, it's very in the case of protein structure, it is sometimes useful to describe nucleic acid structure in terms of hierarchy levels of complexity like primary, secondary, and tertiary structure. So the primary structure of a nucleic acid is its covalent structure and the nucleotide sequence. Any regular stable structure taken up by some or all of the nucleotides in a nucleic acid can be referred to as secondary structure. Now the complex folding of large chromosomes within eukaryotic chromatin and the bacterial nucleotide or the elaborate folding of large tRNA or RNA molecules is generally considered tertiary structure. So the process by which RNAs are formed on a DNA template is known as transcription. In bacteria, mRNAs can contain the protein coding sequences for one gene, like monocystronic, or multiple genes, it is polycystronic. So mRNAs also contain non-coding regions at their ends and between protein coding sequences that may be involved in regulation of protein synthesis. Now, what do we mean by base stacking, and is base stacking possible in single-stranded RNA? So the product of transcription of DNA is always single-stranded RNA. Now, single strand tends to assume a right-handed helical conformation dominated by base stacking interactions. So, in the structure, the bases are shown in yellow over here. The structure, the phosphorus atoms in orange, and the ribose and phosphate oxygens in green. So base tracking interactions are stronger between two purines than between a purine and a pyrimidine or between two pyrimidines. The purine-purine interaction is so strong that a pyrimidine separating two purines is often displaced from the stacking pattern so that the purines can interact. So that is how strong the purine-purine interaction is. So after self-complementary sequences in single-stranded RNAs produce ever more complex secondary structures. So base pairing in RNA matches the pattern observed for DNA. Namely, G pairs with C and A pairs with U for the occasional T residue in some RNAs. So one difference is that the base pairing between G and U residues is allowed in RNA, where complementary sequences in two single strands of RNA pair with each other. The paired stranded RNA duplexes are antiparallel as in double helical DNA, and the predominant double-stranded structure is an A form, right-handed double helix. So the B form double helix has not been observed for RNA. So strands of RNA that are precisely complementary over long regions of sequence are uncommon. So breaks in the regular A form RNA double helix caused by mismatched or unmatched bases in one or most strands are common and result in bulges or internal loops. Hairpin loops form between nearly self-complementary palindromic sequences. Then if we talk about RNA secondary structures, the potential for base paired helical segments in many RNAs is extensive, and the resulting hairpins are the most common type of secondary structure in RNA. So the extensive secondary structures of an RNA is illustrated by the M1 RNA component of the enzyme RNase P of E. coli that functions in the processing of tRNA. This enzyme also contains a protein component. The two brackets in the figure here indicate additional complementary sequences that may be paired in the three-dimensional structure, and the blue dots that you can see in the figure indicate non-vascular G bases space pairs, and you can see that they are only allowed when the pre-synthesized RNA strands anneal together and are not inserted by RNA polymerases and RNA strand across a G in a template. Okay, so this is the potential of RNAs to form secondary structure. Now, extensive secondary structures act as starting points for the folding of an RNA molecule into its precise three-dimensional structure. So, other contributions are made by hydrogen bonds that are not part of the standard. Okay. So what of these include bands involving 2' hydroxyl group of ribose and the oxygens of ribose phosphodiester bonds, the three-dimensional structures of an alanine tRNA of yeast, a hammered ribozyme from certain plant viruses, and a cell splicing intron of mRNA from the ciliated protozoan Tetrahymena thermophila in the figure are shown here in the figure. Okay, so accordingly, our question was about purines and pyrimidines. So we discussed all of that. So now we don't really have any doubt regarding this type of questions. What is the reverse complementary sequence of the following DNA sequence? ACC TTGA. Anybody? This is very easy. Anybody try to answer?
It's TGGAO.
Correct. From which direction? Correct. So if you take from 3' to 5', it will be TGGAACT. The answer is correct. Only that you have to write from 5' to 3'. So that is how you'll write the answer. Okay. So yeah. So DNA is composed of complementary strands. We know that we have discussed so much about that, and we know how G and C, they have triple hydrogen bonds between them, whereas A and T have double hydrogen bonds between them, and the parallel and the complementary strands of DNA are anti-parallel to each other. Correct. So base pairing is determined by the hydrogen bonding. As I said, for adenine and thymine there are two hydrogen bonds, whereas for guanine and cytosine there are three hydrogen bonds. So what are the forces stabilizing this DNA double helix? Hydrogen bonding, obviously, stacking, hydrophobic interactions, and some amount of electrostatic forces as well. So if you see, this is the structure of bDNA. Here the sugars are in the 2' endo conformation. The bases are anti-conformation. Bases of alpha helical twist of 36 degree. Now the right-handed helix helical axis passes through base pairs. The planes of bases are nearly perpendicular to the helix axis. Okay, so this is the major schematic of a bDNA. A DNA can deviate from ideal Watson-Crick structure. Helical twist basically ranges from 20 to 40 degrees Celsius. 42 degrees, sorry, not Celsius. Helical twist ranges from 20 to 42 degree, whereas propeller twist will range from 10 to 20 degree. So that is the angular deviation from ideal Watson-Crick structure. What are the major and minor groups? If you can check, this is the major group and this is the minor group. Okay, so this is what we see in the major group because it has three hydrogen bonds, and the minor group will see a different thing. Now, hypothetical situation, if there's the two groups would have similar size of DR residues are attached at 180 to each other. So that is why the major and minor group demarcation is very important. This is a comparison of A, B, and Z DNA. You can take a look at it for some time. Regarding the shape, rise per base pair, helix diameter, skew sense, diagocytic bond, base pairs per turn of helix, pitch per turn of the helix, tilt of the base pairs from normal to helix axis, major groove and minor groove. Okay. Now, what are the structural characteristics of DNA associating DNA protein recognition? So, major group of DNA contains sequence dependent patterns of H bond donors and acceptors. Okay. So there can be hydrophobic interactions via intercalation, ionic interactions via phosphates. So, DNA separation can be done using agarose electrophoresis, or even people can use PH gel, which is polyacrylamide gel electrophoresis. So DNA are negatively charged, they will migrate towards the anode. Okay. So we know about DNA endonucleases also. DNA endonucleases are basically some enzymes that cleave internal phosphodiester bonds, resulting in 3' OH and 5' phosphate ends. And some endonucleases can even cleave randomly. Okay. Type 2 restriction endonucleases are highly sequence specific. So they are found in bacteria where they are used for protection against foreign DNA. So this is the regular sequence of some common mutation in the nucleus. Okay, we'll not go into too much detail. So coming to the summary, genetic information is stored in DNA. Obviously, we know that DNA is a double-stranded biopolymer containing repeating units of nitrogen base, deoxyribose sugar, and phosphate. DNA can be arranged in three types of duplexes, which contain major and minor groups A, B, and Z. DNA can adopt several topological forms. There are enzymes that will cut DNA, ligate DNA, and change the topology of DNA. And the human genome contains about 3.2 billion base pairs. Individual differences observed as about 1 per thousand nucleotides. So, if this is the sequence given to us, the reverse complementary sequence of this DNA sequence is this. Correct that what we discussed. Ideally, it should be from 5 prime to 3 prime, but it should be T G G A T. Sorry, yeah, T G G A C T. Yeah, that will be how it should be, and that is how, what she like what has been shown over here. Okay, so this question is also done. We'll move on to the next question. Which of the following are true about water? At high dielectric constant, low surface tension, high specific heat capacity, any product of water is 10 to the power minus 7, and it being a polar solvent. So I'll give you a few minutes time to think about the answer of this question. One second. Did someone, am I audible? Yeah, okay. So I'll start the question. I'll give you a few minutes to discuss about this, think about the question, and let me know if you know the uh, answer when I'll show the slide. Yeah. Which of the following are not true about water? Anybody? Anybody? You can try to think about the answer. Okay. Is there anything else that is false? Anybody else? Think about it. We can think for some time, then only we'll start discussing. Think about it for some time. Anybody? Okay, 1 second, give me a minute. There is again a lagging issue. 1 second, I'll reshare. Give me a second. Let me share again. Yeah, hope I'm audible. So we'll discuss each of the points and see if they're true for water or not. So low surface tension is false for water. Ionic product of pure water is 10 to the power minus 7 is also false. Okay. So what is water basically? Water has a higher melting point, boiling point, and a heat of vaporization than most other common solvents. And these properties result from the strong attractions that occur between water molecules in both liquid and solid ice states. If you see here in this table, this is if you compare the melting and the boiling points of everything with water, you can see that water has a higher melting point, higher boiling point, and heat of vaporization than most of the other common solvents. So water is a polar solvent because hydrogen and oxygen atoms have substantially different electronegativities, that is different affinities for electrons. So because electrons are shared unequally, the OH covalent bonds are dipolar and the partial positive and negative charges occur in hydrogen and oxygen. So this feature plus the fact that there are two unshared pairs of electrons are located within the sp3 orbitals of oxygen atoms, they create a basic net dipole moment. Now hydrogen bonds and non-covalent interactions occurring between the H atom of a dipolar molecule such as water and the unshared electron pair of another atom, that is oxygen or nitrogen. So these bonds represent the primary way in which water molecules interact with themselves and many types of biomolecules. The strength of hydrogen bonds is on the same order of magnitude as the thermal energy of an aqueous solution. Okay, so this hydrogen bonds are relatively short-lived. The term flickering clusters basically is used to describe the short-lived groups of water molecules interlinked by hydrogen bonds in liquid water. Now, each water molecule in ice forms 4 hydrogen bonds to its neighbors, creating a regular tetrahedral crystal lattice. So, in liquid water, each water molecule forms hydrogen bonds with an average of 3.4 other water molecules. So, due to the more expanded crystal lattice structure of ice is less dense than liquid water and ice floats on water. So, hydrogen bonds account for the relatively high melting point of ice and boiling point of liquid water. Now, water molecules are not the only molecules that form hydrogen bonds. In fact, hydrogen bonds readily form between an electronegative atom, hydrogen acceptor, usually oxygen or nitrogen, and a hydrogen atom covalently bonded to another electronegative atom in the same or another molecule. So, because carbon is about the same electronegativity as hydrogen, hydrogen atoms covalently bonded to carbon atoms do not really participate in hydrogen bonding. Now, alcohols, aldehydes, ketones, and compounds containing NH bonds all form hydrogen bonds with water molecules and tend to be soluble in water. Molecules such as sugars and amino acids, which contain these functional groups, they commonly are quite soluble in water because of the stabilizing effects hydrogen bonds have on interactions between the solvent and the solute. Okay, so if you see, this is a bit the hydroxyl group of an alcohol and water, carbonyl group of a ketone and water, between the peptide groups in polypeptides, between the complementary bases in DNA. So the hydrogen bond directionality, and hydrogen bonds are highly directional in that strength depends on the proper alignment of the interacting atoms. So the best alignment will occur when the orbital containing the unshared electron pair of the acceptor atom is in line with the covalent bond between the donor atom and hydrogen. So directionality will confer bonding specificity as with the Watson-Crick hydrogen bonds between the bases of double helical DNA. Okay, and will result in very precise three-dimensional structures for nucleic acid and protein molecules. Now, polar biomolecules that dissolve easily in water are referred to as hydrophilic. Now, non-polar biomolecules that do not dissolve appreciably in water are called hydrophobic. So, amphipathic biomolecules have significant amounts of both hydrophilic and hydrophobic structure. So, like hydrophobic biomolecules, they tend to associate when placed in contact with water molecules. And examples of these three classes of biomolecules are shown in this kind of a table. See the glucose. So, if you can see, polar can be basically glucose, glycine, aspartate, lactate, glycerol. Non-polar can be these. Amphipathic can be phenylalanine, phosphatidylcholine. Now, what are the ionic interactions occur between cations and anions? So, these bonds are non-directional and strength depends on the distance of separation according to 1 by r square. So, strength also depends on the medium, the dielectric constant, and is less in polar than non-polar solvents. So, water is effective at screening the electrostatic interactions between dissolved ions because it has a high dielectric constant, a physical property that reflects the number of dipoles in a solvent. So the electric constant of water is 78.5, whereas the electric constant of benzene is 4.6. Now again, ionic compounds such as sodium chloride are readily dissolved in water. Now water molecules can hydrate and stabilize the Na plus and Cl minus ions. If you can see over here, they basically weaken the electrostatic interactions between them and countering the tendency to associate in a crystalline lattice. So resulting increase in entropy of the system is largely responsible for the ease of dissolving NaCl in water. The solvation spheres of water molecules surround ions in solution. So water molecules orient so that the negative ends of their dipoles contact cations and the positive ends can contact anions in solution. So that note the random orientation of the water molecules. Now, hydrophobic molecules such as hexane and the non-polar portions of amphiphites such as long-chain fatty acids lack polar functional groups that can interact with water molecules. So, this results in a highly ordered cage-like shell clathrate of water molecules immediately surrounding the non-polar molecule. So, suspension of a hydrophobic substance in water is thermodynamically unfavorable due to the decreased entropy of water molecules in the cage-like shell. Now, the hydrophobic effect and the term hydrophobic interaction refers to the entropy driven aggregation of nonpolar molecules in aqueous solution that will occur to minimize the ordering of water molecules with which they are in contact. So this is not an attractive force but rather a thermodynamically driven process. So amplified such as detergent molecules or long chain fatty acids, they can form molecular assemblies that are called micelles, micellar solution micelles, wherein the hydrophilic portions of the molecules will be in contact with water, surrounding water, and the hydrophobic portions will be sequestered away from water in the interior of the assembly. So the hydrophobic effect drives the formation of membranes and contributes to the folding of proteins and the formation of double helical DNA. So the molecular interactions displace bound water molecules. So a somewhat ordered layer of water molecules will occur around all solutes, even polar ones, in liquid water. And in enzyme substrate interactions, ordered water molecules are displaced due to binding. So the entropically favorable release of these water molecules will provide a thermodynamically push uh, towards formation of the enzyme substrate complex. If you see, there is a ordered water interacting with substrate and enzyme. So disordered water displaced by enzyme substrate interaction. So enzyme substrate interactions stabilized by hydrogen bonding ionic and hydrophobic interactions. So what are van der Waals interactions? Van der Waals interactions are bonds between fluctuating induced dipoles within the electron clouds of interacting molecules. These bonds can occur between non-polar or polar molecules. Van der Waals bonds are extremely dependent on the distance of separation between the molecules and are significant only when the electron clouds of the molecules are just touching. So values for van der Waals radii and covalent radii of some common elements are listed. Okay. So you can see the non-covalent interactions and macromolecular structure features. So non-covalent bonds are weak electrical bonds between molecules. They can be either hydrogen bonds, ionic electrostatic bonds, and van der Waals interactions. They're typically 100-fold weaker than covalent bonds, and their stability is only slightly greater than thermal energy in biological systems. So, but still, they still play important roles in protein and nucleic acid stabilization because they're collectively strong. All of them together collectively they will be strong, and the hydrophobic effect will drive molecular interactions but is not a non-covalent bond per se. Okay. So about the binding of water molecules to proteins, so structural analysis indicates that some water molecules are tightly bound to proteins, and this is illustrated for hemoglobin here. So these water molecules have different properties from those of the bulk of water of the solvent. Then certain proteins, tightly bound water molecules play roles in catalysis and roles in ligand binding. Now, what are the qualitative properties of H2? Solutes alter the vapor pressure, boiling point, melting point, and osmotic pressure of aqueous solutions. So, in all these cases, these physical properties change due to the fact that the concentration of water is lower in solutions than it is in pure water. So, the effect of solute concentration on the qualitative properties of water is independent of the chemical properties of the solute and depends only on the number of solute components dissolved in the sample of water. Okay, so if you talk about movement of water across the plasma membranes, you have talked about osmosis, which is basically the diffusion or the movement of water across the same permeable membrane in response to osmotic pressure difference between the across the membrane. So most isolated cells have a high concentration of dissolved solutes within their cytoplasm. So they are prone to damage by water uptake induced cell lysis. The terms isotonic, hypertonic, hypotonic refer to the relative concentrations of solutes in a cell cytoplasm and the concentration of solutes in the water in which the cell is suspended. So water will always move towards the side of the membrane with higher solute concentration. Okay. So this is a water molecule. So the octet rule states that there are 4 electron pairs around an oxygen atom in water, and these electrons are in 4 sp3 orbitals, and 2 of these pairs covalently link to hydrogen atoms to a central oxygen atom, and the 2 remaining pairs remain non-bonding or lone pairs. So water geometry is a distorted tetrahedron. So the electronegativity of the oxygen atom induces a net dipole moment, and because of the dipole moment, water can serve both as a hydrogen donor, bond donor, and acceptor. Okay, so there are strong dipole interactions that arises between an acid and a base. They can form between hydrogen, oxygen, nitrogen, and fluorine. So hydrogen bonds are strongest when the bonded molecules are oriented to maximize electrostatic interaction. So ideally, the three atoms involved in the hydrogen bonding should be in a line. Water can serve both as an H donor and an H acceptor. So up to 4 hydrogen bonds per water molecule gives water its anonymously, high boiling point, high melting point, and unusually large surface tension. So the low surface tension option was incorrect. Water has an unusually large surface tension because they can be at least four, like up to 4 hydrogen bonds per water molecule is possible. And hydrogen bonding in water is cooperative, so hydrogen bonds between neighboring molecules are weak relative to the HO covalent bonds. So you can see over here, these are the length of the distance of the covalent bonds, and that is the distance of the hydrogen bond. So water is a good solvent for charged and polar substances, amino acids, peptides, small alcohols, carbohydrates, but it is a poor solvent for non-polar substances such as non-polar gases, aromatic moieties, and all of that. Okay. Now, water dissolves many salts. So there's a high dielectric constant that will reduce attraction between oppositely charged ions in salt crystal. Essentially, the water shorts out the interaction between the two ions, and strong electrostatic interactions between the solvated ions and water molecules will lower the energy system. So entropy increases as ordered crystal lattice is dissolved. Okay. So importance of hydrogen bonds will be the source of unique properties of water, source and function of protein, structure and function of DNA, structure and function of polysaccharides, binding of substrates to enzymes, binding of hormones to receptors, matching of mRNA and tRNA, all of these, the hydrogen bonds importance is very visible. So these are examples of hydrogen bonds. So what are the biological relevance of hydrogen bonds? It can be the hydroxyl group of an alcohol and a water, between the carbonyl group of a ketone and water, between peptide groups in polypeptides, between complementary bases of DNA. So van der Waals interactions have two components. Attractive force depends on the polarizability and repulsive force depends on the size of atoms. So attraction dominates at longer distances, repulsion dominates at very short distances. So there is a minimum energy distance, van der Waals contact distance. Okay, if you see this table, this shows the van der Waals radii and covalent radii of some elements. Not go. What is the hydrophobic effect? It basically refers to the association of or folding of non-polar molecules in the aqueous solution. The main factor, it's one of the main contributors behind protein folding, protein-protein association, formation of lipid micelles, binding of steroid hormones to their receptors, and it does not arise because of some attractive direct force between the two non-polar molecules. Okay. If we talk about solubility of polar and non-polar solutes, you can check out this table where where they have talked about some solubility of some gases in water. So low solubility of hydrophobic solutes can be explained by entropy. So bulk water has little order, high entropy. Water near a hydrophobic solute is highly ordered with low entropy. So low entropy is thermodynamically unfavorable, thus hydrophobic solutes have low solubility. So water surrounding nonpolar solutes has lower entropy. So what is the origin of the hydrophobic effect? For example, if you consider amphipathic lipids in water, lipid molecules will disperse in the solution. Nonpolar tail of each lipid molecule will be surrounded by ordered water molecules, and the entropy of the system will decrease. Okay. So you can see this is schematic of dispersion of lipids in water. Each lipid molecule forces surrounding water molecules to become highly ordered. Now again, non-polar portions of the amphipathic molecule will aggregate so that fewer water molecules are ordered, and the released water molecule will be more random and the entropy will increase. So all the non-polar groups are sequestered from water and the released water molecules increase the entropy further. So only polar head groups are exposed and make energetically favorable hydrogen bonds. So if you see the schematics, these are clusters of lipid molecules on the top. So only the lipid portions at the edge of the cluster force the ordering of water. Fewer water molecules are ordered and entropy is increased. Whereas in the second image, these are micelles. All hydrophobic groups are sequestered from water. Ordered shell of water molecules is minimized and the entropy is further increased. Hydrophobic effects apparently favors ligand binding. So the binding sites in enzymes and receptors are often hydrophobic, and such sites can bind hydrophobic substrates and ligands such as steroid hormones. Many drugs are designed to take advantage of the hydrophobic effect as well. So if you see ionization of water, OH bonds are polar and can dissociate electrolytically. Products are proton and a hydroxide ion. So dissociation of water is a rapid reversible process. So most water molecules remain unionized. Thus pure water has very low electrical conductivity and the equilibrium is strongly to the left. So extent of dissociation depends on the temperature. Now, what is proton hydration? Protons do not exist free in solution. They are immediately hydrated to form hydronium or oxonium ions. A hydronium ion is a water molecule with a proton associated with one of the non-bonding electron pairs. Hydronium ions are solvated by nearby water molecules, and the covalent and hydrogen bonds are interchangeable. So this allows for an extremely fast mobility of protons in water via proton hopping. Okay, so this is the reason why the mobility of protons in water is very fast. You can see hydronium ion will give up a proton and the proton will keep on hopping. Water will accept a proton and become a hydronium ion. We had talked about this. We've talked about pH. pH is a logarithmic scale. I talked about the dissociation of strong and weak electrolytes. Okay, a specific heat capacity. A specific heat capacity is the quantity of heat required to raise the temperature of 1 gram of substance by 1 degree Celsius. Okay, so what specific heat capacity of water is 4.18 joules per gram degree Celsius, and this is the equation for getting the specific heat capacity. Heat transferred into mass into specific heat capacity into change in heat transferred will be mass into specific heat capacity into change in temperature. Okay, now the ionic product of water. So pure water is a very weak electrolyte and ionizes according to the equation over here. Okay, so dissociation takes place to a very small extent. So the concentration of undissociated water molecules may be regarded as constant. Okay, so the product of concentrations of H1 and OH minus at a particular temperature is termed as the ionic product of water. Okay, so the values of Kw will increase with the increase of temperature, that is the concentration of H plus and OH minus increase with increase in temperature. So the ionic product of water or Kw at 25 degrees Celsius is 1 into 10 to the power minus 14, and since pure water is neutral in nature, H plus ion concentration must be equal to the OH minus ion concentration. Okay, so what was our question? The ionic product of water is 10 to the power minus 14 and not 10 to the power minus 7 as was mentioned in the question. So let's go back to the question that we started with. Which of the following are true about water? Has a high electric constant? True. Low surface tension? False. High specific heat capacity? True. Ionic product of water is 10 to the power minus 7 molar? False, because it is 10 to the power minus 14. Polar solvent? True. So these two statements are false, whereas the other statements are true. Okay, we'll stop here today. Does anybody have any question regarding what we discussed today? Anybody? Okay, if nobody has any questions regarding what we discussed today, we can conclude this session and we'll meet again on Monday. Next Monday. Thank you. Thank you, guys. I'll close this session. Thank you.